will try to give you a so-called tour de force through epigenetics, and in particular environmental epigenetics today. And please, Roxana, stop me if I overrun. If I still got 45 minutes, I think I will manage to get through the key messages which I wanted to give you. I first want to give you a very short introduction about epigenetics and DNA methylation. I think some of you might not be too familiar with the subject yet. I will then talk about 4 specific aspects of epigenetics: aging, plus-minus hormone replacement therapy and DNA methylation, effect of smoking on DNA methylation, diet on DNA methylation, and HPV on DNA methylation. And at the end, I will show you how we anticipate to integrate the actual information that we have gathered throughout the last 4 years within the HEAP consortium into a concept which I think has the potential to transform cancer prevention. Well, epigenetics has been something which is quite exciting also from the lay perspective, because it is the interface between the genome and the environment. And the epigenome is so-called the software of your genome, which basically is the cell program. And here the Time said why your DNA isn't your destiny. The Germans said the victory over your genes, and then Darwin was wrong, was said by the New Scientist. So epigenetics is something which has attracted a lot of attention in the past. But it is very complex. It's certainly more complex than genetics, and it consists largely of 4 components, all regulate gene expression and gene activity in a heritable fashion, heritable from cell to cell. And it basically includes histone modification, as you can see here. You can't see the mouse. The non-coding RNA, as well as DNA methylation. You see these bubbles coming off the DNA. This is 5-methylcytosines, if you see it in this context. And this is what we are talking about. We are talking about DNA methylation. Very brief introduction. You see here the sequence of the DNA. Only sites which are in the CG dinucleotide context, so these are these things that are labeled in red, can be methylated. And they can be in the methylated or in the unmethylated version. So if they are in the unmethylated version, they are called cytosines. If they are in the methylated version, they are called 5-methylcytosines. And you see this covalent modification of the DNA makes all the difference. What does DNA methylation do? It regulates in a heritable way gene expression via chromatin modification, and this has a massive impact on cell differentiation, on a cell's identity, really important in terms of cancer, and cell function. And so a lot of different environmental factors have shown to impact on DNA methylation, and thereby are likely to modulate cells' function and cells' identities. And obviously, what we want to do is to see how these factors impact on the epigenome, on the individual epigenome, and that's what our subject is. Because if you understand how these factors impact on the epigenome, then it might really be important, or it might lead the way how you can identify specific individuals with certain risks. The key challenge, and this is something which is quite important, there's 2 things. One is obviously the environment that Alison has very nicely demonstrated, which you can capture with these novel devices. But what matters at the end is how the individual responds to these specific factors. And in terms of cancer, and this is the key message of today's talk, in terms of cancer, because cancer is largely an epithelial disease, there is obviously also blood-borne cancers and immune cell-associated cancer, but for the majority of cancers, these are out from epithelial cells. And in terms of environment shaping the epigenome of cells, it matters which cells you study. So epithelial cell epigenome is the key here, and this is the key challenge that we face, because the majority of population-based cohort biobanks are blood, and very, very limited resources exist, one of which is the Swedish cervical biobank that actually have biobanked epithelial samples, in this case, cervical samples. So this, I just wanted to give you a flavor. What does the epigenome do? It indicates the past. So how have we been exposed? Age obviously is something that is also the past in some way. HPV exposure, antiprogestins, for instance, is something that interests us. It basically reflects the present. Currently, we are working on utilizing DNA methylation in epithelial samples to identify individuals with breast or ovarian cancer, potentially use this to predict the future risk for it, and then obviously indicates the future disease risk, in this case, for instance, endometrial cancer and cervical cancer. Cervical cancer is the subject with risk prediction, which has progressed the furthest, and there will be a paper, which I'm not talking today about, be published in the next few days, all based on the Karolinska Biobank. So let me tell you what I'm liking to do today. I'd like to focus on these 4 aspects: age, smoking, diet, and HPV, and how it impacts on carcinogenesis, and what we basically, and this is literally all the work that we have done within the HEAP program, how it impacts. So let's start with age. And you see at the top here, the ThinPrep system. This is basically the main source that we have used. These are samples that have been obtained with these brushes from the cervix, and then the brushes with the cells on it have been placed into these 20 milliliter preserve site. These are methanol-based fluids, and these ThinPrep samples have been mainly used for our purpose. So that's why I've mentioned them here. One aspect that we have specifically highlighted, which has also been quite under-researched and underestimated, is that these samples, in particular, when you extract the DNA, because the DNA is also extracted from cells that have undergone cell lysis due to apoptosis, necrosis. So you're basically not just isolating the cells from the vital cells, but you're isolating the entire DNA from the sample. And when you do this and you use a DNA methylation-based reference epigenome and use deconvolution algorithms, you basically can assess what the different proportions of cell types in a given sample are. And you see here typical examples. You see that about half of the cells are epithelial cells, about 50%, and the remaining are neutrophils, monocytes, and other immune cells. So this is something which is really, really important to keep in mind. We basically use these samples and utilizing the chronological age of the donors to develop a DNA methylation signature, which could, in this independent validation, almost exactly predict the chronological age of an individual. We call this the Widschwendter clock. But we noticed that different components, epithelial components and immune components, are differently contributing to this age. And we wanted to assess, can we determine a signature which reflects the epithelial age and the general age and the immune age? And what we've basically done is we've identified CpGs, as you can see here on the horizontal axis, which only where age only, or that the methylation changes only correlate with epithelial cells, and on the vertical axis where the CpGs only, or the age of the CpG age only correlates with the immune components. And so by identifying and then combining these specific CpGs, we devised 2 specific signatures. One, which is here under B, the epithelial age, which basically is purely based on CpGs, which change as a function of age in epithelial cells. And on the right-hand side is the immune clock. What we mainly were interested in, what is the relative epithelial age, which is basically the difference between the epithelial age and the general age. And now let's jump a bit because we don't have too much time, but a key message is, you see here, individuals who either took hormone replacement therapy, the red ones, or sorry, the blue ones, or individuals in women who did not take HRT. And what you see on the left-hand side is that healthy controls have, when they take HRT, they have a reduced relative epithelial age, indicating that when they take hormones, that the epithelial age is being reduced, so almost rejuvenated. And these were healthy controls, but when we assessed women who eventually developed breast or ovarian cancer, we did not observe this rejuvenation effect. And when we split up the signature into the general clock and into the epithelial clock, you can see here that in the controls under panel B, the epithelial age is decreasing, whereas in the panel C, women who had developed breast cancer in the future, this epithelial age is not coming down. This is an extremely interesting finding, but obviously would benefit strongly from further validation, in particular assessing the longitudinal assessment. How long does it take for a woman to show this rejuvenation effect or don't show this rejuvenation effect? And can we actually utilize this information to individualize and risk adjust hormone replacement therapy? So let's see whether this video actually works. The noise or the thing doesn't work. We've just done a video. You can see the actual text at the bottom. It really just shows the way how we've developed this. This is DNA methylation shown in a different context. We've done the relative epithelial age as we've shown you before, and we've basically utilized this information and found that in the hormone replacement therapy slows down the epithelial aging, but only in women on the left-hand side, on the left-hand side, which do not develop or did not develop breast cancer. In women on the right-hand side who did develop breast cancer, we did not see this phenomenon, which is quite interesting. But again, it would really benefit from either a prospective assessment and or assessment in a large cohort of women, where we also have the information of HRT exposure as well as the information about the outcome. So what we really need is large prospective observational studies, including longitudinal epithelial biobanks, in order to guide personalized risk-adjusted hormone replacement therapy. So this about age and DNA methylation and how it can be modified by hormone replacement therapy. The next aspect is smoking. Smoking is obviously one of the most dangerous environmental exposures, which is best studied in terms of cancer predisposition. So it's a very nice model to assess. What you see here is a busy slide, and I don't have a pointer, but you see two phenomena which I think is really important. One is obviously you have the tissue at risk. Tissue at risk for the lung is the bronchial epithelium, and you're not able to get access to the bronchial epithelium without the general anesthesia and the bronchoscopy. So you're going to need to have a surrogate tissue. And the concept of the surrogate tissue is absolutely essential for what we want to get across. It basically is tissue which is easy accessible, like the epithelial cell of the cheek in this case, and has a similar context. It's an epithelial cell and it's not an immune cell. So this is something which is really important. Surrogate tissue, in this case, easy to access epithelial cell. And the second aspect that we wanted to get across here is obviously you can enumerate or you can quantify the exposure by saying how long have you smoked, how much have you smoked. You know, you can quantify this in pack years, but each individual is different. And the bottom line here, sorry, I can't show this, but maybe I can here. So, yeah. So this here is basically what other lifestyle factors has this person been exposed? What other environmental has this person been exposed? And then genetically, or by whatever means, the absorption, the metabolization, the germline genetic background, and the cell programming, all is different in different individuals. So this individual component can only be captured if you basically assess the sort of common denominator, which eventually is the DNA methylome. And that's the beauty of studying DNA methylation. It does not only record yes or no, exposed or not, but it basically records the individual response to this exposure. So what we've done in a quite large study, we did want to assess the long-term impact of smoking by epigenetic changes, but this has been studied by a large number of people, including our team in the past, but it basically was only focused on blood. Again, people were looking into blood because the source is available. These EWAS, epigenome-wide association studies, have only been done in blood, but there was no information on changes in epithelial cells. And no studies, and this is another shortcoming, has actually studied the direct exposure. So if you study direct exposure, you see the Buccal cells, but obviously we know that smoking also triggers other cancers like cervical cancer, for instance. And so we wanted to assess how are the changes in other cells which are not directly exposed, blood being one, or the cervical epithelial cells being the other source. So we studied cell type specific changes in diverse samples and evaluated the associations with smokeless tobacco, something which is quite important as well, e-cigarettes, and lung cancer. So we wanted to utilize the epigenome in order to say, can we almost foresee the future? Can we assess how new exposures, like the exposure to e-cigarettes, might potentially impact future disease predisposition by using this as a snapshot at this point in time without having to wait 2 or 3 decades until the actual disease, which might be triggered by this new exposure, is showing its face. So to cut a long story short, we've assessed different aspects. We've looked into epithelial cells, so we've assessed buccal samples, blood samples, cervical samples, and wanted to address the proximal, means the direct exposure. Proximal means direct exposure in the buccal sample, so distal means blood or cervical exposure. And so I go through this a bit faster. All of this has already been published. And then we've identified 4 signatures, and we wanted to assess these signatures in different contexts, in e-cigarettes, snuff tobacco, changes in lung cancer tissue. How do these signatures changes in lung cancer tissue compared to the normal exposed lung tissue? And how is it associated in the actual progression of in situ carcinoma into invasive carcinomas? So the main findings are shown now. So let me guide you, and this is quite complex, in particular with the speed that I'm presenting this data. So these are the 4 signatures that we had derived. You see here the epithelial hypomethylation. So you see the blue ones is CpG sites, which are less methylated in the exposed people. And you see these are epithelial cervical, these are epithelial from the buccal. In both, they are hypomethylated. Then you see the immune hypomethylated. So immune hypomethylated means in the blood hypomethylated, but also in the immune cells that are in the buccal or the cervical cells. And you see here, they're all hypomethylated. Then you see the distal epithelial hypermethylated signature, which is more methylated in the cervical samples here. They are also a bit methylated in the epithelial samples of the cheek swabs. And then this is the most important signature. This is the proximal epithelial hypermethylated signature. And you see a very strong methylation in the epithelial cells of the buccal samples, meaning this is epithelial cells which have been directly exposed. So keep this in mind, proximal epithelial hypermethylation signature. I basically put these boxes around it so it's easier for you to focus. We've also done some pathway analysis, and so the most important, the proximal epithelial hypermethylated pathway shows some involvement in growth signaling. One of the aspects that we wanted to assess in independent sets, obviously all the data that come now are independent from the training set, is how malleable are these signatures, which is quite important. So you see here, the signature that is epithelial hypomethylated, you see the red ones, those are the active smokers, and you see the orange, the ex-smokers, and you see the blue ones, they are never smokers. And you see here the immune cell proportion. So on the left-hand side, those are samples where you only have epithelial cells, no immune cells. On the right-hand side, you have samples that only almost exclusively exists of immune cells. And you see the big difference here in the smokers, because the signature is called epithelial hypomethylation. You see a low methylation compared to the non-smokers and also compared to the ex-smokers. So the ex-smokers almost went back to the normal situation. So quite quick going back to the default. And then if you look into the proximal epithelial hypermethylation signature, again, focus on this aspect where you see the high proportion of epithelial cells, high methylation in the active smokers, but then still quite a lot of methylation in the ex-smokers. So the the way back is far more difficult for this really important signature. And here you see the non-smokers. And when you basically discriminate between the smokers and the non-smokers, or the smokers and the ex-smokers in this case, so smokers and ex-smokers, you see that the hypomethylation, there's a big difference, basically meaning that the ex-smokers cannot be discriminated from the non-smokers for this when applying this specific signature. But when you actually apply this signature, which is the epithelial hypermethylation, then you don't see a big difference between the smokers, which obviously can be discriminated to with 100% sensitivity and specificity, and the actual ex-smokers. So this is something which is quite important, that the going back is slow, and we are currently doing a study where we assess how slow it goes back. The next study obviously is to understand, do these signatures have the capacity to discriminate between the normal lung and the lung cancer? Because that's what is something that you would expect if this is functionally relevant. We don't show functional relevance in a pure form, but we can do some approximation by comparing the methylation level of these signatures in the normal cancer, in the normal tissue. So this is the normal tissue in blue, or the cancer tissue. And we have lung adenocarcinoma and we have lung squamous cell cancer. Just focus on the two panels here for the time being. And what you basically see, and I focus down now in the interest of time only on this proximal epithelial hypermethylation signature, you see that for both lung cancer types, you see a very strong increase of methylation in the lung cancer tissue compared to the normal lung. And you also see here, I don't know whether you can see this at the bottom, these are 3 samples. One is a control tissue from women or an individual without cancer. And this is a carcinoma in situ lesion, which upon follow-up has regressed, so has not progressed to cancer. And this is a carcinoma in situ lesion, again from the lung, but has eventually progressed into a cancer. And you see that this proximal hypermethylation region, which signature, which has been developed based purely on studying DNA methylation in healthy individuals in cheek swabs, is able to highly discriminate between the progressors and the regressors, respectively, those that have no cancer. So in other words, these signatures do seem to be somehow at least favoring the progression or the clonal evolution of cancer cells. What we were then interested in is, how do these changes appear in people who are exposed to other sort of cigarette smoke, which is shown here. So these are from a study that has been done in Bristol from our collaborators. These cigarettes and these are e-cigarette users. The blue ones are e-cigarette users, and the red one are smokers. And you see the area under the curve when you compare to non-smokers. So the comparison is to non-smokers. Area under the curve basically plots the sensitivity against the specificity. 1 basically means perfect discrimination. 0.5 means no discrimination. And what you see here is the signature that we had shown before, this epithelial hypomethylation, which is unaffected in the e-cigarette smokers, but is affected in the smokers as predicted. But when you then look into the signature that we are particularly interested, which is the proximal hypermethylation buccal epithelial cell signature, you basically see that e-cigarette smoke exposure is basically doing almost exactly the same thing as smoking. We can talk about this, the details of the study later, so I just skip this. Again, this has already been published. So this is something which is quite important. If the exposure to e-cigarettes leads to the same epigenetic changes that you see in the normal smokers, and we show that this signature is higher in the cancer, we would expect that this has some pathogenic relevance. Again, we can't say anything about the pathogenic mechanism. The snuff users is pretty much the same. Snuff also causes this hypomethylation, but again also causes this hypermethylation in the actual buccal signature. And so we've done a very small study, and this again shows and highlights the lack of data and the lack of samples we have. These are data from the NSHD 1946 birth cohort. This is a cohort who had been established back in March 1945. 5,000 pregnant women have been approached. All children have been born in March 1946, have been followed up throughout their entire lifetime. And we had the opportunity to assess BACRE samples from 150 individuals and blood samples from 150 individuals when they were 52. From 52 onwards, we observed them for another 20 years, or 22 years, and you see the area under the curve is 0.71, but obviously the confidence intervals are extremely wide because the number of events was obviously low. But this again gives some indication that this epithelial signature in BACRE cells is also predicting the risk for future cancer. But again, it basically really shows the lack of samples and the lack of data that we have, and we really need large, long-term, well-annotated epithelial biobanks to study this. The third aspect, and I keep it relatively short, where's the time? Okay, it's okay. I keep this relatively short, is a study that we've done generically within the HEAP program, which aimed to understand how intermittent fasting is impacting on our health, in particular on our epigenome. So we have comprehensively evaluated the systemic impact of 6 months of intermittent fasting. Intermittent fasting, in the case that we've defined, it is 16 hours fasting, so no complete caloric restriction, and only 8 hours. You can either skip the breakfast or you can skip the dinner. And then we've done this for 6 months. We've had a randomization into only intermittent fasting. So this was prospectively randomized, only intermittent fasting or intermittent fasting plus a ketogenic supplement. Ketogens, ketone bodies, substances that the body generates in order to replace the lack of glucose to nurture the blood, the brain, and the heart. So this is something that we've added on artificially in one group. And then we did conduct a baseline assessment, and then 2, as you can see here, 2, 4, and 6 months assessment. We are now following up 12 and 18 months, but we are reporting currently these 4 time points. And we've basically assessed everything that you can almost imagine, which can be assessed non-invasively, including the epigenome, including the metabolome, including the microbiome, including pulmonary health, including metabolome, etc., etc. And I only give you a flavor of the findings that we've made. You see, obviously, not everyone was as compliant as we expect. This is the low compliance group, this is the median compliance group, and this is the high compliance group. So you see the color code, and you see here the body mass index. The body mass index, even in the low compliance group, is decreasing. It's obviously much more decreasing in the medium and in particular in the high compliance group, as you can see also here, as the delta BMI compared to the baseline. You see the fat mass that we've assessed using the bioelectric impedance analysis. You see here the low compliance group, you see the medium compliance group and the high compliance group. Obviously the fat proportion goes down. You see the VO2 max, which is a very important metabolic parameter, which is assessed respirometry, based on respirometry. You see an increase, which is highly significant. You see that the good cholesterol, the HDL, goes up, and the LDH, lactate dehydrogenase, which is a parameter of cell death, is decreasing. So this is really just a random assess or a random showing of some of the clinical data, focusing on DNA methylation. Again, just pulled out to show you some of the data. We did assess the impact of intermittent fasting on something which is called the GRIM age. GRIM age is something that others have developed. It basically shows the age that reflects really bad outcomes. So if you have a high GRIM age, chances that you are dying soon is relatively high, in a simple terms said. So what you basically see is here the baseline M0, then M2, M4, and M6, and you see a significant decrease when you assess this in blood. So for this, we had to use blood because the original signature had been developed in blood. So this is something which was quite satisfying. We then also looked into the global methylation. So it's quite complex. In a nutshell, the global methylation is usually higher in good cells, normal cells, and it's lost in cancer cells. And specific promoter-associated methylation is increasing in the cancer compared to the normal tissue. So in terms of global methylation, you want to have a higher methylation. And this is what we've shown here as well. Based on Backel samples, you see that the methylation levels based on this L1, so this is a repetitive element, is increasing, which is quite interesting as well. And then we've developed and published in the past a parameter which is called the WIT-QTBC marker, which utilizes DNA methylation, which is associated with genetic sites which predispose to cancer. That's why the QT is in the front of this. And it basically is decreasing as well as a function of time and exposure. And it's significantly lower after 6 months. So this is really just one of the few findings. One aspect which specifically caught my attention is the following. We always hear that the immune system and the sort of granulocyte effect is adversely impacting on cancer, on carcinogenesis via these different pathways. You see here the E-cadherin pathway, you see the NF-kappa-B pathway, etc. So these granulocytes, in particular the myeloid cells, the majority of them are granulocytes, release factors which impact on the biology of epithelial cells, and these epithelial cells are then more prone to become cancer. And so what obviously our technology allows, due to this deconvolution algorithm, it allows us to measure DNA methylation and assess the proportion of all granulocytes in a sample, irrespective of whether you could have seen them under the microscope and you only see them if they are in a cell context, or whether they already disintegrated. So this we've taken the entire DNA methylation from the entire sample using this deconvolution algorithm. And you see here again months 0, 2, 4, and 6. And you see this intermittent fasting effects in blood. You basically don't see really any difference. The granulocytes were in this sort of 0.6 or 60% range in the peripheral blood. When you then look into the Backel samples, in the Backel samples, we saw relatively low granulocytes, but even in these low granulocyte containing samples, you see that with increasing fasting, you see a decrease in these granulocytes, which I found quite exciting. And the most exciting thing is when you look into the cervical samples, that the original, that the baseline samples was quite variable and quite high. And then months 2, months 4, and month 6, the actual proportion of granulocytes dropped significantly, which we don't know the mechanism about. It could be via altering the microbiome. It could be via altering the cell adhesion. This is something which we'd like to understand further. But nevertheless, I find this quite an exciting finding. You hear more about the follow-up studies, which is the LIFE study and the SUN study. LIFE study has further assessed these lifestyle changes based on 700 volunteers now, all assessed 8 months. More tomorrow from Sandra and the other team. We also want to make sure that these data are publicly available, which poses specific challenges in terms of containing identifiable, potentially identifiable information, in particular linked to exposure. So we have this, we have generated, or we are generating this open access infrastructure and scaffold, where you have some data which are purely open access without, and then you have controlled access, and then you have a restricted protected data which is only available for GDPR compliant countries. So this is something that we are currently establishing. This is the final part of this, but again, this should lead to sharing the data, the samples that we have generated, in particular in these settings, which are quite challenging to achieve. The last bit, and I really keep this very, very short, is the effect of the HPV on the DNA methylome, something which came a bit as a surprise. This is already published, but I just wanted to highlight this as well. HPV obviously impacts also on DNA methylation, but this was yet unexplored, and we wanted to assess. So this is a bit blurred. Anyway, I'll talk you through this. It was not well studied so far. And so we utilized samples from, again, the Karolinska Biobank to assess the impact of HPV on DNA methylation. So we've compared samples who have been HPV negative to samples who were HPV positive but cytology normal, so no abnormality, and derived the signature which we call the Widschwendter test. And then you basically see this is normal samples, so this is a validation set. Normal samples HPV negative. These are normal samples, cytology normal samples HPV positive. These are then samples which eventually turned out to have SYN1 as an underlying cause, and this SYN2. So you see that they all show this high methylation or this high signature. But then, and this came as a massive surprise, when you then look into samples that eventually turned out to have an underlying SYN3+, so either SYN3, which is a pre-invasive lesion, or invasive cancer, the HPV signature went back to normal. So you would have expected that it went further up, but it did exactly the opposite. It went down to the normal level. So there was a massive difference between normal cytology SYN1 and SYN2, and it almost fell down, which came as a big surprise. So we wanted to get and assess this further, and we've looked into this, and we found two things in a nutshell. One is shown here. It basically shows that the apoptosis index is dropping. So we've developed an apoptosis index independently on samples, on cellular samples, developed this DNA methylation signature, which is reflective of apoptosis. And you basically saw that in the SYN3 samples, the apoptotic index is low. And you see this correlation of the HPV index with the apoptosis index. So high apoptosis index means high likelihood of apoptosis, high with HPV index. Low means here. And here you have got all the SYN3 samples. And we've also assessed the replicative age, just to cut the long story short. Replicative age can also be assessed using DNA methylation at specific sites. We call them polychrome group target sites. And the picture that emerges, again, this is just the picture that emerges, is that if you have epithelial cells in the cervix, which have undergone a high replicative age due to chronic infections, due to other factors, then these cells, usually progenitor or potentially stem cells, which have undergone a high replicative age, have accumulated DNA methylation, which eventually prevents them to undergo apoptosis would they be infected with HPV. So this is, I think, quite an exciting mechanism which has emerged out of these data. But obviously, we would need to assess this in a much bigger context and potentially utilize this. So bringing everything together, how do we think long-term, possibly beyond my lifetime? How do we try to bring this all together? So in the center is the individual. This is something which is, at least in my environment, something that we have always to be reminded. And then we basically have this P4 medicine. This, I think, is quite a nice guidance. It's very simple. First, you have to identify the risk. So this is what we do in the cervical cancer screening, HPV. So this is very well established, but it has to be established also for other diseases. So we need to do a risk assessment. Part of what I've shown basically aims to achieve this. The second thing is, if we go for primary prevention, we need to apply specific primary preventive measures. A lot of those are available. Lifestyle is one. Specific targeted prevention is another thing. We are, for instance, triple-negative breast cancer. We're using mifepristone, a progesterone antagonist, to achieve this. And there's a variety of other aspects. And then the stuff which is really, really important, you're going to need to have an intermediate surrogate measure that allows you to assess how and whether this primary preventive measure is actually working or not. You can't wait until the person developed or did not develop a cancer. So we need to have something that allows us to measure the efficacy, and DNA methylation is good for both the prediction as well as the personalization in order to measure the actual efficacy. And then obviously most important, you need to ensure that it is simple and easy so that a lot of individuals, in the majority, can actually attend. So if you do need to stick needles into something or do some painful or difficult things, the majority of the population will not attend. So participatory is the last but probably most important aspect. And so the way we see this, I promise you, is the last slide, is we have the participant, we have the general practitioner, or we have prevention nurses. I would rather do this in a very low community-based setting in order to ensure high attendance. And then you basically measure specific aspects. I would basically strongly argue that DNA methylation is a very good means to identify the risk. And based on the risk, you then do specific primary preventive measures or secondary preventive measures. Secondary preventive measures is screening, intensified screening, etc. And then you basically have specific registries, which has been quite well established. You have a digital prevention file, possibly something that one should think of. Obviously, we involve a lot of digital media. We have in our lifestyle studies done a real sort of telemedicine approach where we have had apps that allowed us to communicate directly and provide some of the guidance, or actual programs like the resilience program that we have in the lifestyle study. Sandra will talk to you about this tomorrow. And then obviously this has to be repeated. So this is a loop where you then for the next cycle assess these features and see whether the primary preventive measure has actually achieved what is meant to achieve, or whether the primary preventive measure has to be adjusted. So this is the big vision that we see, and this is why we're so fascinated by DNA methylation and epigenetics, because we see the real potential of this to achieve this. Big thanks to primarily my team. You all know Chiara. Chiara has done a lot of this work, but also to other people who have been involved in this: Charlotte, James, Elisa, and Elena. And you will hear tomorrow, as I said, from the other team. Big thanks to our long-term collaborators. A lot of it would not have been able to achieve this without Joachim's and his team's support. So really big thanks. This has been a fantastic opportunity. And then in this context, also the Medical University of Graz, and big thanks to the European Union who made all of this financially happen.